How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, they're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello, I'm your host, Keir Deneen, and you're listening to DNA Today, where we discover new advances in the world of genetics. Today, we're learning about long non-coding RNAs, also called lncRNAs for short, and I'm joined by Sharon Turcotte, who has done research on these molecules. Welcome to the show, Sharon.
Thanks for having me.
Yeah, I really appreciate you coming on and just explaining this molecule to us and the significance of it and how much we know, how much we don't know. So a little bit about Sharon for you listeners. She just finished an internship at Jackson Laboratory for Genomic Medicine, located at the UConn Health Campus in Farmington, Connecticut, because I don't like talking about Jax enough. Now I'm bringing someone on from Jax. I'm a big Jax fangirl. So she focused on studying lncRNAs and is going to share her experience with us today on the show. So can you start by explaining what an lncRNA is? It's probably a good base point. To me, it seems kind of like a bit of an oxymoron, because when I hear RNAs and people that are kind of don't have as much genetic exposure, they hear RNA, it's transcribed from the coding part of DNA, the part that's making a protein. So what is an lncRNA?
Well, a lncRNA, or like link is how people typically pronounce it, so
okay,
you don't have
good
to say
know.
LNC a million times. It gets hard.
Yeah.
lncRNAs are, like you said, they're a bit of an oxymoron. Typically, the dogma in biology is DNA, RNA, protein. But lncRNAs do not code for proteins whatsoever, or as they say, they have low protein coding potential. But what they do is actually really various depending on the lncRNA themselves. The research on lncRNAs is still evolving. So right now it's basically just defined as an RNA molecule which is over 200 base pairs long and performs various functions. So they're defined as a heterogeneous class of molecules, and that's basically a fancy way of saying they do a bunch of these different things, and we're kind of giving them this haphazard definition, but we're still studying them. So, for example, lncRNAs can perform protein binding functions, so they can carry proteins to certain places on the DNA to kind of be involved with gene expression. lncRNAs can bind specifically to certain sequences of DNA. They can act as conformational switches. That also has to do with gene expression. They can act to be a scaffold for proteins and carry specific proteins to other places in the nucleus. So they're really doing a variety of functions, none of which the mechanisms are really, really well understood yet, but they have a lot to do, again, with gene expression. So I would say if I had to pin down one function, that would be the biggest one for lncRNAs.
And we're doing so much research on this because we just don't know a lot. It's kind of like we think they do these kind of things, and you're saying that through your research, you've really found that gene expression is one of those main functions that it does. And for those that don't remember what gene expression is, that's when we are controlling the genes that are expressed. That kind of makes sense, but that means which genes are expressed into proteins, and the proteins are kind of what we're made of. So through your research, what have you found the general structure of a lncRNA molecule to be? Is it similar to an mRNA or a tRNA?
First, I'd like to clarify that we are not, well, it's not we anymore, but at Adam's lab at Jax, they're not studying the specific structure or function of lncRNAs in general, but we are looking at them from a phenotype standpoint. And I'm sure we'll get into that a little bit later, but part of research is looking at the literature and looking at what is already known about the thing you want to look more at. So in that regard, before the research in Adam's lab was started in earnest, of course, you have to kind of see what is already known. So to answer your question, the general structure of a lncRNA is more similar to tRNA than mRNA. mRNA, messenger RNA, tends to be linear and, goes to the ribosomes to become proteins. tRNA is involved in that process as well, and, it's kind of famous for having that T shape. It binds to itself and makes that very unique shape that serves a specific function to build proteins. lncRNA also kind of binds within itself to make a variety of shapes depending on the length of the lncRNA, and can be anything from like a T shape to like a crazy, like all these multiple arms of it. And so it's really has this vast variety of shapes depending on the lncRNA itself or its function.
So does the shape, you mentioned size, is the structure really dependent on the size, or you can have, say, a 300 base pair lncRNA molecule and it can be a variety of shapes?
I would say certainly the length of the RNA molecule is going to determine what its shape can be. But even something that is 300 base pairs long, and maybe you have 4 different link molecules that are 300 base pairs, depending on what those letters are, the C's, the T's, the G's, the A's, you're going to get different shapes because it will bind differently to itself within that molecule. Their functions are better understood, obviously, but there's a whole host of lncRNAs that haven't even really been identified yet. And so their structure hasn't been cracked. Their functions haven't been cracked in terms of the study. But, like, for example, lncRNAs have... I'm sorry, could you... I'm not sure if I actually answered your question.
I'm trying to remember now. I was, like, focused on what you were saying. I'm trying to remember what my question was. I guess I was talking about gene expression and...
You said that they did a variety, like, there was a variety of ways to...
Right, I said there's a variety of...
expression.
Oh, right, okay. Do you want me to restate the question, or should we just skip the question?
You can restate the question, and then maybe we can go back.
Okay, sounds good. Just gonna make a mental note. We're 10 minutes in. Okay. So lncRNAs have a lot of different structures, and a lot of times structure determines function. With gene expression, there's a lot of different ways to express genes and have those genes turn into proteins. Do we know lncRNAs' specific roles in gene expression, or are we kind of like, they do something in gene expression, we're not sure what?
So as a heterogeneous molecule, lncRNAs in general have different functions depending on the lncRNA gene. Unlike other aspects of biology that are well defined, lncRNAs are kind of in the beginning preliminary understanding phase, and as such, they have a lot of variety within the class itself. So one lncRNA may affect methylation around the DNA. Another may have another function. So it might recruit a protein instead that affects chromatin, for example. So it really just depends on which lncRNA you're talking about to determine its specific function.
And can it activate gene expression and inactivate, or is it on one side more than the other?
It is involved in both upregulating and downregulating certain genes.
Okay, cool. So gene expression, obviously a big one. Is there any other big functions that we know of lncRNAs, or we're really questioning, or there's a lot of hypotheses out there?
For the most part, lncRNAs are expressed within the nucleus specifically. because they're not being coded into proteins, so they're not really, seeing the ribosomes in the cytoplasm as much. So for the most part, I would say it's safe to assume they're mostly involved in gene expression and having to do with the DNA at this point. Whether or not we will find another kind of function for them is yet to be seen. But, the golden rule in biology is there's an exception to every rule.
Definitely. And that makes sense if it's really primarily in the nucleus, because most of what's in there is DNA. So kind of pairing that logic together, that makes sense. If we have lncRNAs that are mutated or absent in the body, what effect does this have? Do we know?
Oh, yeah. There's been multiple studies linking correlations with mutations in the lncRNA genes themselves with a variety of diseases, which is one of the reasons Adam's lab at JAX is looking into them. You've got everything from heart disease, multiple cancers, autoimmune diseases, neurological degenerative disorders. All of those have been correlated with single nucleotide mutations in the DNA.
And knowing that it affects these types of disorders and diseases, do we see any possible therapeutic treatments that could be designed knowing about lncRNAs or even using lncRNAs?
I think part of the therapy, at least our hopes for the future, is being able to either upregulate or downregulate certain genes. And that's kind of as a general statement, not specifically to lncRNAs. It's something that's really kind of far down the line in terms of future applications for specific therapies. But for lncRNAs specifically, we have to figure out what they do and how they work first before we can use it kind of as a medicine. But, for example, one of the goals of Adam's lab at JAX is if we can find a certain DNA or a certain lncRNA that plays a major role in asthma pathogenesis, then if we can turn that gene off, it might be possible to ease people's suffering within that disease if we can, identify that gene and then deliver some kind of therapy that is going to either upregulate or downregulate that specific lncRNA.
So you're really talking about a lot of epigenetics happening, things that are controlling the expression levels, and also gene therapy, which has kind of been a hot topic, but I think recently I've seen it more in the news of we're closer to actually using gene therapy.
Yes.
On a more widespread account. CRISPR technology is super hot field in genetics, especially like the last 3 years, really. It's exploded. Have you used any CRISPR systems in your work? It's something that gene therapy is really looking at right now.
Yeah, I definitely want to touch on the point you just made, is that gene therapy is getting closer and closer and closer. And that is in part due to the discovery of Cas, the CRISPR-Cas9 enzyme. Before they didn't really have a super reliable way of editing any genome, let alone the human genome. So CRISPR-Cas has become kind of the go-to, the gold standard in gene research now. And that is true of Adam's lab as well. We are using a modified Cas enzyme called DCAS9 with CRAB. And basically, it's the CRISPR enzyme itself that has a different protein kind of stuck to it, which disables its nuclease region, which basically means that it, instead of cutting the DNA like Cas9 usually does, it no longer cuts the DNA. So we kind of like dull its scissors, if you would want to use that analogy. And then instead of cutting, it modifies the chromatin to make the DNA coil more tightly around that region and therefore cannot be accessed by molecules, which will lead to protein building.
So not only can CRISPR-Cas, so the Cas is the protein that usually when we read in literature does the cutting of the DNA to inactivate, but here you're using it in a different way. You're actually not cutting, as you said, you're kind of dulling the scissors. I like that analogy, that you're dulling the scissors to, are you changing the structure, or are you changing the methylation for the, this is going to get a little complicated here, for the histone proteins to be, for the chromatin to be more tightly wrapped around the histone proteins to inactivate?
On the modified Cas enzyme, like we said, the scissors have been dulled, so they no longer function. The crab protein that is kind of stuck to this new modified Cas enzyme is working on the chromatin, on the methylation, to kind of cause it to coil more tightly around specific regions of DNA, which have been kind of driven there or guided there, as you might want to say, by the gRNAs or guide RNAs that we design in the lab.
And when you're doing this, are you targeting the genome or the lncRNAs?
That would be both.
Both.
So we, specifically in Adam's lab, we are looking for lncRNAs that play a role in asthma. That's like the long-term goal. But in order to do that, we have to kind of test them one by one. So we design certain guide RNAs one by one and use them within a modified Cas enzyme to turn off those genes, to make them inaccessible to ribosomes and messenger RNAs and polymerases and all those things. They can't get to them anymore, so they can't express the proteins, express the genes. And then basically we look at changes in the phenotype of the cell.
That's so cool when you just think about all that you do to do this and just how vast CRISPR technology is. And from reading literature on my own, I've read that it's a lot easier to use than other gene editing technologies. Would you say that's true, that the CRISPR technology is kind of easier than maybe other gene editing technologies and other technologies in the lab?
Yeah, certainly. That's pretty much why everybody's using it now, or at least trying to use it, and why everyone is so excited about it. I can't speak specifically to the older technologies. I'm not as familiar with them, but I do know as kind of a general rule, they were less specific and more prone to errors. They might like bind off target or cut off target, or just not be as efficient as Cas. Cas is much more efficient than what we had previously, much more reliable, and does not bind off target nearly as often as other technologies did previously.
It's using that guide RNA to bring it right where it needs to most of the time, right?
Yes, it is very highly specific, so you can really drive it exactly where you need it to be.
Certainly an exciting technology that I'm very much interested in looking where it's going to go in the future. Now, if we take a step back, we've talked about you using the Cas proteins to do this gene editing and messing with the gene expression. But how do you study the lncRNA molecules? What cells are you looking at? What's the process of getting to the point we just talked about?
Oh, it's a long workflow in general. I think to more seasoned scientists, they are probably like, oh, that's fine. But for me, who is relatively new to the research end of things and to science in general, it seemed, very complex. There's a lot of different steps that need to happen before we put anything in any cell. Kind of the first step was deciding which link genes we were going to look at in general. So before I came to the lab at Jax et al., my supervisor and my mentor, Ellen Elliott, looked at RNA-seq data just to see which link genes were being highly expressed in the cells that they ultimately want to study, being human T cells and white blood cells. So they look at that, and they have a way of telling, okay, which ones are being used the most within our white blood cells that we want to study. And they compile a list of 20 or 30 link genes that you then have to look at from there. After you've decided what genes you're going to look at, then you have to figure out, okay, how are we going to look at them? And from there, it's about designing those guide RNAs that you're going to use in Cas ultimately. Basically, phase 1 is finding which link RNAs are highly expressed. That's going to give us our stepping off point from where we're going to go from there. After you figure out which ones you want to look at, it's a matter of confirming their expression in actual cells. So you build guide RNAs, you make a plasmid, you get that in bacteria, you isolate them, make sure that everything you want in the plasmid is in the plasmid, including those guide RNAs, including the information to build the Cas enzyme. And once you have that, you can put that information, that little plasmid of DNA, inside actual cells to try and turn off gene expression. And once you've done that, we use a transient transfection which basically means a temporary transporting of these new genes we're kind of putting inside just to turn off genes. It sounds kind of silly to say that. Like, we're putting in new genes to turn off genes, but we need to tell the cell how to build the Cas enzyme. We need to tell the cell how to build the guide RNA that's going to go and find those link RNA genes. We need to tell the cell how to do that. We do that using a plasmid. So we put the plasmid inside the cell, and then hopefully we can see that those genes the expression of those genes have been changed. And from there, it's a matter of kind of like the data gathering stage. The last stage of the experimental workflow is, did the expression of my genes change? Did the phenotype of my cells change? And from there, you can kind of start to quantitate things and qualitate things and see what happened.
So how do you know if your genes were successfully, if the plasmid that you were inserting into cells, if it actually was incorporated?
Yeah, that's an excellent question. There are several safeguards kind of in the experimental workflow to make sure that we did the thing that we wanted to do. So one of them is within the plasmid that we make for the cell, we have a GFP tag, which is basically kind of a glowing, like a phosphorescent tag, so that the cells will actually glow when excited under a certain wavelength of light, so that we can physically see, okay, these cells didn't have this GFP tag before. If our plasmid gets into the cell, then the GFP tag gets into the cell. And then when we look at it under a certain kind of microscope, they should fluoresce green. So it's a really clear way of seeing whether or not your plasmid has gotten physically into the cells. And you can like take nice pictures and stuff, and it's like, oh wow, they're there. You can see green.
And who doesn't love glowing molecules or glowing cells? That's pretty cool,
right?
I remember in high school, one of the first genetic kind of labs I did was inserting the GFP gene into plasmids and then putting that into cells and seeing them light up, and it was just the coolest thing because you're like, you just did that. Like,
yeah, you totally did that.
Yeah, so it's a very cool thing, and it's something that a lot of scientists use, which is why I thought it was important to ask you, because that's something that is very widespread throughout genetic studies and all of that. So you've mentioned a couple names at JAX. Who worked with you on this project? You've mentioned is it Williams lab, you said? Yep,
Adam Williams is the PI, our principal investigator. So, obviously he's the head honcho. I worked very closely with Ellen Elliott, who was my supervisor and mentor, and basically taught me everything that I'm telling you right now. So hopefully it's right. Thank you, Ellen. I also worked with Dinesh. I don't want to try to say his last name and then mess it up. Mm-hmm. And also a nice woman named Magali. So those were Oh, and also Maureen was our great secretary. So I worked with all of those people pretty much every time I was there. And they all have their own kind of certain projects within the research as a whole. Ellen's project, which was, in turn my project, was figuring out how we were going to knock down these genes, these link genes within the cells.
And did you collaborate with any scientists outside of JAX? I mean, other than reading literature and seeing what was out there about lncRNAs?
Me personally, no. Whether or not Ellen or Adam will do that in the future is an open question.
Cool. Yeah, I know I was doing a little research myself on lncRNAs and stumbled across rnacentral.org, which obviously I didn't go through the whole thing, but it's a non-coding RNA sequence database that I thought was pretty cool, and I'll definitely look more into if listeners are interested in learning more about non-coding RNA sequences and what's out there right now and what's being discovered. That was a cool finding. Now, I kind of want to ask you, in terms of being a student, how you learned about this opportunity and kind of the impact it had on you and that nature.
Sure. I actually kind of ended up like in a really lucky situation, right place, right time kind of deal with me. My advisor at Middlesex Community College, Michelle Kraskowski, she was working on a grant through the state of Connecticut called the Genomics Workforce Consortium. And it just so happened that on the panel that was working with that was also Walter Nakanecki, who is the kind of PR person for JAX laboratories. So they were working on this grant from the state to fund internships, basically, for people who might graduate and then enter the genomics workforce. So they knew they were getting this money, and Michelle knew that she had a student who needed an internship, and Walter knew that there were spaces at JAX. So it was just kind of like, okay, we know that these two things exist. It's going to be funded. There's going to be space. So we need students is basically the question there. And then it was just a matter of applying, because obviously I was not the only candidate for using the grant, and then, interviews, resumes, stuff like that.
That's cool. Yeah, it's awesome that you were in the right place, right time, as you said, that that was able to come together for you, but also kind of lesson for students here to make sure you're talking to an advisor and you're close to them and kind of letting them know what you're interested in, what's going on in your kind of career as you're starting it. Because probably if you didn't do that, you wouldn't end up with the internship, right?
Right, exactly. If I, hadn't been keeping close tabs on my advisor, I might not have heard about it. And also, just so people out there don't feel like, oh, I have to be lucky. How do I be lucky? Like I have no control over that. But you can also just be your own advocate, and you have to look also for yourself. Like before I got into this grant program, I knew that JAX had two separate internship programs because I had done an internship the previous summer, and they took us on a tour of JAX. And also, on JAX's website, they have advertised. They have an academic year internship program, and they also have a summer internship program.
And that's up in Acadia. Beautiful.
Yeah, like it's both are great opportunities. The summer one is especially, it's very prestigious and highly sought after. But like, my point is that you have to, as a student, be your own advocate, and you have to be looking. Like we live in an age where information has never been as easy to get to. So even a simple Google search, like internships in insert city name here or insert state name here, you're going to find stuff. There's stuff out there. It's just a matter of connecting yourself to it and kind of being the squeaky wheel, like the squeaky wheel gets the grease. So if you're asking your advisor every couple of months, hey, have you seen any internships up? Or just don't forget, I exist, I'm looking for this thing, or, whatever, you will probably, quote, be lucky.
Yes, you can only kind of be lucky if you're setting yourself up to be lucky almost.
Yeah, just don't be afraid to look and don't be afraid to ask. That's like my best advice to people.
Yeah, definitely. You see something cool, like talk to the person doing the research, say, hey, I found this really interesting, had a question about it, and kind of getting your foot in the door that way.
Absolutely. And even my mentor, Ellen, she wrote to Adam herself, like to get her job at Jax as a postdoc fellow. There wasn't like even a job listing. She knew she was graduating and knew she wanted to be in the area and was proactive about looking up, okay, who's doing research in that area in my field. So she happened upon Adam's name and some papers that she liked, papers that were in her area of expertise. And she just reached out to him with an email saying, hey, I just graduated. I'm looking for work. I've read your stuff. I'm doing this. I'm interested in this. Do you want to work together? Do you want to collaborate? So I think it's important for people to understand, like, if you are proactive in looking at someone else's work, seeing if you're a good fit, and then kind of reaching out to them in just like, hey, can we collaborate kind of way, you might be surprised what doors will open for you that you might not have even known were there.
Definitely. I couldn't say it better myself. You very nicely put that all together, and just go out there and be your own advocate. Definitely. Well, that wraps up today's episode of DNA Today. You can follow on Twitter at DNA Podcast or website, dnapodcast.com. Any questions for me? Probably more Sharon for this episode. You can email in at info@dnapodcast.com. I will forward those on to her and get a response back for you. So thank you so much, Sharon, for teaching us about lncRNAs. I've definitely learned a lot today.
Thank you for listening.
Yes, and thank you for listening, listeners out there. You can join me next week to learn and discover new advances in the world of genetics.